Thank you very much, Sven. Ladies and gentlemen, I think we're all familiar with the notion a picture is worth 1,000 words, as seeing is believing. And this not only applies to our daily lives, it definitely applies also to the natural sciences, as we just heard. Because I don't think it's a coincidence that the beginning of the natural science in history very much coincides with the invention of the light microscope. Because with the light microscope, we were able to see for the first time that every living being consists of cells as basic units of structure and function. And with the light microscope, bacteria were discovered and mitochondria as subcellular organelles. However, we learned at school that the resolution of a light microscope is fundamentally limited to about half the wavelengths of light, which typically amounts to about 200 nanometers. And if you want to see smaller things than that, we have, such as viruses, for example, we have to resort to electron microscopy. Now, electron microscopy achieved a much higher spatial resolution, 10-fold, 100-fold, 1,000-fold, in fact, down to the size of a single molecule. And therefore, the question comes up, why do we care for the light microscope and its spatial resolution now that we have the electron microscope? Now, the answer to that question is given in the next slide, where I made a little experiment. I counted actually the numbers of papers in this journal where a light microscope was used and where an electron microscope was used. And now you see which of the two systems won. It's still the case today that light microscopy is the most popular microscopy technique in the life sciences. And that is for two strong reasons. The first reason is that light microscopy is the only way by which you can look inside a living cell in 3 dimensions, or even living tissue. So it's minimally invasive. But there's another reason. If you want to look in a cell, usually we are interested in certain proteins or other biomolecules, and we have to make them distinct from the rest. We have to highlight them, because to light or to electrons, all the proteins look the same. And in light microscopy, this is easily possible by attaching a fluorescent molecule to the protein of interest, for example. And that fluorescent molecule has a very interesting feature. It has 2 states, an energy ground state and an energy excited state. And if you shine light on it, for example, green light, it can absorb a green photon, and then what will happen, the molecule will be raised to a higher lying state, then its atoms will wiggle a bit, that's why we have these vibrational substates. Then after about 2-3 nanoseconds or so, the molecule relaxes by emitting a fluorescent photon. Now, because some of the energy of the absorbed photon is lost here in the wiggling of the atoms, the fluorescent photon is redshifted in wavelengths, it's orange in here. But that's good, because then we can easily separate it from the excitation light, so from the light with which the cell is illuminated, and that makes fluorescence microscopy extremely sensitive. In fact, it can be so sensitive that you can detect a single molecule, as has been discovered by our co-laureate W. Myrna. However, if a second molecule, a third molecule, a fourth molecule, a fifth molecule comes closer together than to the first, than 200 nanometers, we cannot tell them apart, because they appear in the microscope as a single blur. So resolution is about telling things apart, telling small features apart and distinguish them and detect them as separate entities. And now you can imagine that because fluorescence microscopy is limited to 200 nanometers, a lot of information is lost if you look with a fluorescence microscope into a cell. So anything that is below the scale of 200 nanometers appears blurred. So therefore, if we manage to come up with a light microscope or a fluorescence microscope that has a much higher spatial resolution, that would have a tremendous impact in the life sciences in particular. Now we have to understand first why the resolution of a light microscope is usually limited. In simple terms, it can be explained as follows. The most important element of a light microscope is the objective lens. And the role of this objective lens is nothing but to concentrate the light in space, to focus the light down to a point. However, because light propagates as a wave, it's not possible for the lens to concentrate the light on a single point. Rather, the light will be smeared out here in the focal region, forming a blob of light that is at least about 200 nanometers wide and about 500 nanometers along the optic axis. And this has a major consequence. If we're having several features falling within this range, of course, they will all be flooded at the same time with this light, with this excitation light. And hence, they will produce signal at the same time. And if you try to detect this, the signal with a lens, and put it onto a detector, the signal produced by these features within this 200 nanometer blob will be confused. So if you look at this one, it produces this blob, and the other one that blob, and so on. And so no detector will be able to tell the signal here apart, no matter if it's a photomultiplier, the eye, or even a camera. Now, the person, as we heard, who discovered that diffraction poses a serious limit on the resolution was this man, Ernst Abbe, who lived at the end of the 19th century, and he coined this barrier in an equation that is named after him. It's saying that in order to be separable, two features of the same kind have to be further away than the wavelengths divided by twice the numerical aperture of the objective lens. And you can find this textbook basically, this equation basically in any textbook of physics, of optics, but also in textbooks of biochemistry and molecular biology because of the enormous relevance of light microscopy to this field. But you will also find it on a memorial that was erected in Jena, in Germany, where Ernst Abbe lived and worked, and there it is written in stone. And this is what scientists believed throughout the 20th century. Not only they believed it, it also was a fact. For example, if you want to look at cytoskeletal features in the 20th century, this was the type of resolution that you got. But now, today, we get this, and this is a standard. And so what I'm going to talk about is, I would like to show you how this transition was made actually from the standard previous resolution to this type of resolution. Now, it started out in the late 80s. I was a student in Heidelberg in those days. And I did light microscopy, so I, of course, was familiar with this equation. And I said to myself, how does it come? This equation was coined in 1873, but now we have 1990. So much new physics actually evolved in the 20th century. So many new phenomena were discovered. I had to learn so much for my examination. There should be some phenomenon, at least one, that could be used to overcome this diffraction barrier. Well, I understood that it won't be just by changing the way the light is propagating, the way the light is focused. It must have something to do with the major discoveries in the 20th century. Quantum mechanics, molecules, molecular states, and so on. And for that reason, I started to check my textbooks again in order to find something that could be used to overcome the diffraction barrier in a light focusing microscope. And one day I put it down in writing, so the idea was check out the properties of fluorophore, state properties and so on, or maybe there's a quantum optical effect that could be used to overcome this barrier. And so one day when I was not very far from here in Orbu, in Turku, just across the Gulf of Bothnia, on a Saturday morning, I opened this textbook and stumbled across this page, and that this was a page about stimulated emission. All of a sudden I was electrified. And now I explain to you why I was so electrified. The problem, as I said, the lens concentrates the light in space, but not better than 200 nanometers. And all the features are flooded simultaneously with excitation light. We can't change that. Sure, we can't change that. But maybe we can change the fact that all the features that are flooded here with excitation light are in the end capable of sending light to the detector. Because if we manage to keep some of them dark, the dark state to be precise, such they are not able to send light to the detector, we can separate the bright state molecules from the dark state molecules. In this case, we can tell this one from that one apart, or that's the top view. So the idea was to keep molecules in a dark state. You see, the state is the essence. Now you will see, or say, are there dark states in a dye? Well, let's have a look at an energy diagram. There is the ground state, as I explained, that's the excited state. And for the molecule to emit fluorescent light, it has to be in the excited state. So this is the bright state, but the ground state is, of course, the dark state. Because if the molecule is in the ground state, it won't emit light. So there is a dark state, that's the ground state. Now guess what stimulated emission does? It does nothing but making ground state molecules, dark state molecules. And that was the reason why I was so excited. I found a way to make dark state molecules just with normal fluorophores. And now you can imagine how the microscope works, STAD microscopy. Well, this is the lens, of course, and we have a sample and a detector. And then, of course, we use a beam of light for getting molecules from the ground state to the excited state, so making them bright. And of course, this light will be diffracted here. We'll get this blob of light of about 200 nanometers. And all the signal that is produced here will be going actually to the detector from all the molecules. But now, we use a beam of light that induces the stimulated emission, makes dark state molecules. And the idea is now, of course, that to push the molecules, in the case they get excited, instantly back down to the ground state so that the molecule is not capable of emitting light because it has to assume the ground state instantaneously. Now, the physical condition for doing that is that the wavelengths here of the stimulating beam is redshifted. It has a lower photon energy so that we don't excite molecules, but couple the molecules from this state back to this ground state. That's one condition, but that's not the only condition. There's another condition. Of course, we have to make sure that there is a red photon at the molecule that really pushes the molecule down. Most photons pass by because there is a finite interaction probability of the photon with the molecule. But if you have a certain threshold intensity, you can be sure that it's at least one photon that kicks the molecule down and makes it instantly dark in the case it gets excited. So what you see here is the intensity of the red beam, and this is the probability of the molecule to still assume the excited state, the S1. And now you see after a certain threshold intensity, you've turned it off. You can put as green light as much as you want, but it won't be able to emit. And now the game is easy. We just convert this beam such that it doesn't form a pattern in here, but a ring. And then we can turn off these molecules, as you can see, and here, of course, the ring or donut will become weaker and weaker, and here it is ideally 0. So here we won't be able to turn them off. But let's assume now that we want to see just this fiber. So we have to turn off this fiber and that fiber. What do? Well, we can't make the ring smaller. That's also limited by diffraction. I would say that's not possible. But we don't have to do that. We just have to shut off molecules because this game is about states. And so what do? We make the beam strong enough so that the molecules here are essentially turned off because they're confined to the ground state. Because even now, even here, in absolute terms, the intensity is beyond the threshold. Now we're there. Only here the molecules are allowed to emit, and we can separate the signal from the neighboring fibrous signal. And now we can take pictures, sure. We can move the beams across the specimen, and now we can separate this fiber from that fiber and that fiber, because the molecules are forced to emit sequentially in time, because we play this on-off game. And then, of course, we move on and so on. I'm just highlighting it again so that you understand it very clearly. This is the excitation region, but only this subclass of molecules are allowed to emit. Those here are kept in the ground state, effectively. Whenever you make a check which state they are, you will always find them in the ground state. And we move on, and then, yeah, I'm putting back the donut so that it's clear that I'm using the donut, of course. So, and this turned out to work. Now, this is a standard recording of something that you cannot really see what it is. But now, I'm using slat microscopy, and I'll see that because the resolution is higher, roughly by an order of magnitude, we can clearly discern actually what's imaged in here. So these are nuclear pore complexes, and as a result of the high resolution, it's about 10-fold better than previously. You can see that this nuclear pore complex consists of about 8 molecules as molecular subunits, and you have this 8-fold symmetry coming out very clearly. The comparison with the standard resolution looks like this, and then of course, that's almost no comparison. So, if you have this increase in spatial resolution, of course you get new information. There's no doubt about that. And I would like to show you an application where something new was learned with this microscope. So this is an application which was done in collaboration with virologists, with people interested in HIV. Now, viruses are about 30 to 150 nanometers in diameter usually. And of course, you want to image them with a light microscope. No way, you won't see any protein distribution on them. It'll just be a blur. Now, HIV is about 140, and in this study, the scientists were interested in finding out how this protein called ENF is distributed on this HIV particle. Normal recording, you cannot see anything. High-resolution recording instead, in this case, you see that it forms patterns on the HIV, and what has been found out in this study is that the mature HIV particles, so those that go to the next cell, infect the next cell, have their ends in a single place. So that's a requirement in order for HIV to be very effective. So that's a new insight that was gained as a result of high-resolution imaging. Of course, a strength of light microscopy is that you can image living cells. And this is an example of video rate recording stat microscopy. These are synaptic vesicles in an axon of a living neuron. You can see how they move about and learn about their dynamics and what were their fate in time. And that's important to be able to image living cells. But a live cell imaging at the extreme, so to speak, is this one. Here we open the skull of a mouse. It's an anesthetized mouse, and look into the brain of the mouse at the molecular layer, in the visual cortex. Now, this is a transgenic mouse, so some of its neurons expressed a fluorescent protein, and that's why we can see this neuron quite clearly in here. The rest of the brain tissue is dark. And now we can take sequential recordings and see that actually Those are the receiving ends of the neurons. These are the so-called dendritic spines, and they also slightly move. It's worthwhile zooming in. As you can see in here, this is the spine neck, and this is this cup-shaped receiving end. And the message here is that this sub-diffraction resolution imaging, STAT, for example, allows you to see these tiny morphological changes. And I'm very confident that in the not-too-distant future, we will be able to image the proteins here at the synapse and perhaps even give a visual cue on the mouse and see how this changes actually the protein distribution directly on the synapse, and in the end learn how neuronal communication works and perhaps memory formation. Now, this is just another example. Of course, one can take three-dimensional imaging because you can focus into the brain tissue, for example, and this is a 3D super-resolution recording of actin in a living neuron in a so-called organotypical hippocampal slice. Now, with that, I'm coming back again to the basics, to the resolution. Some of you will ask, what is the resolution that you can get? What's the limit? Is there a new limit? So let's get back to the principle. Well, the name of the game is that we turn off molecules in here. And if we make the region smaller in which the molecules are still allowed to emit, of course, the resolution gets larger. That's clear. So it cannot be described by this equation anymore, very clear. In fact, it must depend on the intensity that is found here on the donut crest and on the threshold intensity, which is a characteristic of the photon-molecule interaction. And so the larger this ratio gets, of course, the smaller the d will get. And so you can imagine that it must be found here in the denominator if you describe it with obviously new equation that is required now. In fact, d scales inversely with the square root of I over Is. So the larger this gets, the smaller d will get. And in principle, of course, in this equation, it goes down to 0. Because if this becomes very large, d goes down to 0. Now here you can't separate the two molecules because both are allowed to emit at the same time, but let's make it a bit stronger, the beam, so that only one molecule fits in. And now you see the resolution limit, so to speak. It's the size of a molecule. Because this molecule is, of course, the smallest entity that you can separate. This is not surprising. We separate by having molecules in two different states. And so it must be the molecule, which is, in the end, the limit of the spatial resolution. So in this cartoon, I'm having two molecules coming very close together. And now we can separate them, because at the time this one is emitting, the other one is off. By the way, if this is totally dark, one photon is enough to know that there is a molecule. Because we know the position of the molecule by the presence of the red photons. They determine exactly where the molecule is allowed to emit and where it's not allowed to emit. Now you will ask, do you get molecular spatial resolution typically, for example, here and there in a cell? Well, with STAT microscopy right now, the standard of resolution is between 20 to 40 nanometers, depending on the fluorophore, depending on the chemical environment, but that's something that is progressive. And I would like to show you that, of course, if you have the right fluorophore that can be turned on and off as many times as you want, you can do better. In fact, there are fluorophores, not organic ones, inorganic ones, that do that already. So these are so-called charged nitrogen vacancies in diamonds. So these are fluorescent defects found in solids in diamonds, and they can be turned on and off almost as many times as you want. And here we managed to squeeze down the region of emission to 2.4 nanometers. Keep in mind, the wavelengths responsible for this is 775. So it's smaller than 1%. So it's a small fraction of the wavelengths. Now, of course, this looks like a proof of principle experiment. To some extent it is, but it's not just that. There's another reason why to do these experiments. These so-called charged nitrogen vacancies currently are very hot candidates for quantum computation, qubits at room temperature, because they have a spin state with a very long coherence time. And also the spin state can pick up magnetic fields, so to speak, at a nanoscale. We have nanosensors in here. But STAT is perhaps the best way of reading out the state. And this makes that perhaps an interesting candidate in the end, maybe for reading out, say, qubits in a quantum computer. Who knows? The development goes on. So I'm coming back again to STAT. I said the name of the game is on-off. This is how we separate it, a bright state and a dark state. Now, once you realize that this is a general principle, not just doing stimulated emission, then you know, okay, there must also be other on and off states in a dye that you can use to the same effect, do the same thing. Well, and so I browsed other textbooks, and I found there are triplet state, dark states, and of course, if you look into your chemistry textbook, you will find there is cis-trans isomerization, as you can see in here. And then there's other options to do the same thing. You may ask again, why do you use these special transitions that are not found in any fluorophore? You need special fluorophores for this. If you have this basic transition here, that's just totally basic, ground state, excited state. Well, there is a very strong reason for looking into these other types of states. Let me show you the reason now. Look at the lifetimes. Here, the lifetime of the state, the excited state is nanosecond. Here, the lifetimes of the state is microseconds. Here, it's milliseconds. So the lifetime of the states increases. So why is this important? Well, it's very obvious. We separate by having adjacent features in two different states. But if the state, one of the states, disappears after a nanosecond, then this difference in states disappears after a nanosecond. And so, of course, you have to hurry up putting in the photons, creating this difference in states and reading out this difference in states. But if you have more time, microseconds, milliseconds, you can turn it off, read it out, turning on, turning it off. It stays there because it has a long-lived state. So you don't have to hurry up putting in the light. And that makes it, so to speak, operational at lower light levels, at much lower light levels. So to be more formal, this intensity threshold IS scales inversely with the lifetime of the states involved. And the longer the lifetime, the smaller is the IS. So you can break the diffraction barrier using this type of transition at much lower light levels. So the IS, the intensity, threshold intensity goes down from megawatts, kilowatts down to watts per square centimeter, 6 orders of magnitude. That makes this transition, of course, very interesting. Here in the equation, IS goes down, and with that, of course, also I goes down because you don't need as much light in order to get the same D. Now, this cis-trans isomerization is particularly interesting because it's found in switchable fluorescent proteins. And so we looked into this very early on to check whether we can use it for a STAT-like recording. And this is, I called it resolved because I couldn't have called it STAT anymore. There's no stimulated emission in it, so I had to give it a different name. Now, the strengths, if you need little light, not only is that you can get high resolution at low light levels, you can say, oh, I'm buying a cheap laser, CW laser, and I spread out the light over a large field of view because I don't need so much. And then I can parallelize the recording, can make many holes at the same time and read out a large field of view quickly. It doesn't matter that I have many. As long as they are further away, that Abbe's diffraction barrier, I can read them out in parallel, project it onto a CCD camera. That's it. Just a few scans in this direction and in the other direction, and I can take a large field of view. And this was done in here. So here, this, say, living cell was recorded within 2 seconds with more than 100,000 donuts, so to speak, in parallel. So again, the key is the molecular transition. So selecting the right molecular transition determines the parameters of imaging. So the microscope and the contrast modality and all the factors are determined actually by the molecular transition that is chosen in here. Okay, then I put up the next question. So what does it take to get the best resolution? Now let's assume you had asked this question in the 20th century. What would have been the answer? I'm sure you know it. Good lenses. Sure, good lenses. Why? Because the separation was done by the focusing of light. And then, of course, you need good lenses to produce the best focus here or the best focus there. And if you cannot produce a smaller focus of light, you're lost. So if you have several features, of course, falling within this diffraction zone, then you cannot do any better. So you're definitely limited by diffraction if you separate by the focusing of light. No way to tell these features apart because everything overlaps in here. So what was the solution to this problem? Not separate just by focusing. Separate by molecular states. In the easiest case, on-off states. Because if you separate by molecular states, you can distinguish the features and can tell it apart, even though they are designed within the diffraction barrier. And now we can tell this one apart from here, from the rest, and can discern it. And you have here a choice of states that I've shown already that you can use to distinguish the features within the diffraction zone. Now, in the methods that I've described, STAT, resolved, and so on, the position of the state, where the molecule is on, where the molecule is off, is determined by a pattern of light. You've seen it in the donut. It clearly determines where the molecule has to be on and where it has to be off. X, Y, Z is tightly controlled by an incident pattern of light. I go to the next. Again, I know the position already. I don't need many photons here because everything is determined here by the position of the pattern. Now the question comes up, how does this compare with a seminal invention by Eric Bettig based on the discovery of W.E. Murner that you can detect single molecules. Well, in the palm concept, there are two fundamental differences to STAT. First of all, it relies on single molecule detection, critically relies on that. That's very clear. Secondly, unlike in the STAT case, in the palm case, the position of the on state is totally uncontrolled, totally stochastic. One molecule pops up somewhere randomly in space. And you create, in this way, this on-off state difference. But since you don't know where you've turned it on, you have to have a pattern of light with which you can find it out. And this is done with a pattern of light that is emitted here by the molecule. Now, without going into the detail, of course, this pattern of light allows you to determine the position with a centroid calculation. The point that I'm making here is you need a bright pattern here to find out the position, just as you need a bright pattern here to determine position. You always need bright patterns of light when it comes to positions. And it's not surprising. If you have just one photon, it goes astray. Here, 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 within the diffraction zone. No way to control where it goes. If you deal with positions, you need many photons by definition, because that's inherent in the diffraction. So, many photons. But the separation, of course, in both cases, is done by an on-off transition. This is how we tell features apart. As a matter of fact, all the super-resolution methods that are in place right now and are useful, they use the separation, they make a separation by transiently placing the molecules in two different states for a time period of detection. Fluorescent and non-fluorescent, that's the easiest to play with. And so this is what has worked out so far. Now you can say, in the 20th century, it was the lenses which were decisive. And the lens makers were the kings. You had to go to them and ask them for best lenses to get the best resolution. But today, no, it's not the lens makers. This is about molecular states. And molecular states is, of course, about molecules. The molecules decide now how good you can image. The molecules decide now the spatial resolution. And molecules, that's not optical technology. That is chemistry. It's now the chemists who make the best pictures in the end. So in a way you can say that this initially physics problem, which diffraction barrier was, no doubt about it, has now been converted into a chemistry topic. And with that, I'm coming to my almost last slide. I couldn't have told you this development without the help of many very talented people who have contributed to this development. Actually, most of them that are shown here are here. I thank them very much from the bottom of my heart for their contributions. And I would like to add that they are still continuing because this field is very, very, very exciting. And I would like to make another point here. Well, the big award now, the Nobel Prize, was awarded to super-resolution fluorescence imaging. But once you have realized that this is the state transition that we have in here, AB need not be on-off in fluorescence, could also be something else, absorption, non-absorption, scattering, non-scattering, spin up, spin down. Then you can also imagine a super-resolution absorption microscope or a super-resolution scattering microscope if you have the right states. And so the story continues, and I'm expecting more of it to come. It has just begun. And this is really my last slide now. You see now, this is Abbe's equation. It was written there for so many years. It cannot explain the fact that we have now a much higher spatial resolution, but we can deal with it very, very easily. We just plug in the square root factor, and now the good news is it goes down to the size of a molecule, so we can have resolution at the molecular scale. Thank you very much.